Now let's take a closer look at nervous tissue. So nervous tissue is also called neural tissue. The nervous tissue can be found in the brain, spinal cord, nerves, and the special sense organs. But almost every organ in your body has some nervous tissue in it. And that's because almost every organ in your body has to be able to communicate with the nervous system. For example, in your skin, you have nervous tissue for detecting temperature and pain. And the nervous tissue there can send signals away from your skin towards your brain and spinal cord. Or for example, you have nervous tissue in your skeletal muscles so that the brain and spinal cord can send signals to the skeletal muscle telling it to contract. When we look at nervous tissue, we see there are two kinds of cells that make up nervous tissue: neurons and neuroglial cells. Neuroglial cells are also known as just glial cells. Neurons are the cell that are going to be doing the electrical and chemical communication of the nervous system. The neuroglia are helper cells, or sometimes they're referred to as nurse cells, that really just sort of take care of the neurons. Now let's take a closer look at neurons. So a neuron has three main parts to it: a cell body, also called a soma, soma means body, dendrites, and an axon, or nerve fiber. The cell body, or soma, takes up the bulk of the cytoplasm of the neuron. It contains most of the organelles, like the nucleus, Golgi apparatus, and rough endoplasmic reticulum. The dendrites are extensions off the cell body that bring a signal into the neuron. So the job of the dendrites is to take signals, usually from other neurons, and bring those into a neuron. Whereas the axon has the opposite job. The job of the axon is to send a signal away from the neuron. Axon starts with A, away starts with A. It's a good way to remember that the axon sends the signal away from that neuron. So in this picture here, this is the nucleus. So this is the cell body, or soma, of the neuron. These small branches here are dendrites. Dendrites tend to be much shorter than axons. Okay? So axons and dendrites are both branches, but dendrites are shorter branches. So these dendrites will be bringing a signal into the neuron. This structure here is the axon, and the axon sends the signal away from the cell body of that neuron towards another neuron. Now we have different types of neurons in our bodies, and we classify them different ways. One of the classification schemes for neurons is to divide them based on the number of extensions that come off the cell body. If a neuron has two extensions that come off the cell body, it's called a bipolar neuron. If it only has one extension that comes off the cell body, it's called a unipolar neuron. So in a unipolar neuron, the one extension itself branches to become dendrites and an axon. But the most common type of neuron in the body, and the one that we're going to use as an example throughout this unit, is a multipolar neuron. A multipolar neuron, as the name suggests, has multiple extensions that come off the cell body. Most of those are dendrites, and only one is an axon. Axons are really important for neurophysiology, so I want to talk about some special structures associated with axons. The axon hillock is actually part of the cell body, but it's the part of the cell body just before the beginning of the axon. And the axon hillock is really important because it contains the trigger zone for a nerve impulse. Axolemma means the cell membrane of the axon. So that suffix lemma means cell membrane. The axolemma has some special properties to it that allow it to conduct a nerve impulse or action potential. We talk more about this in a different video. Axon terminals are the ends of the axon. If you look at the end of the axon here, you can see it has branches. And at the ends of the branches are the ends of the axon, and those are the axon terminals. Axon terminals are really important because they contain structures called synaptic vesicles filled with neurotransmitter. Synaptic vesicles are bubble-like structures that can release their contents via exocytosis, and these are really important because these are going to be used to release neurotransmitter and allow one neuron to signal with the next. Axon terminals form a connecting piece with another neuron, and the place where one neuron communicates with another cell is called a synapse. They don't actually physically touch the two cells. There's a space between them called the synaptic cleft. So when a nerve impulse reaches the end of an axon terminal, it's going to cause the release of the neurotransmitter into the synaptic cleft, and then the neurotransmitter will go and communicate with that other cell that's a part of that synapse. So taking a closer look at a synapse, again, like I said, this is where one neuron communicates with another neuron. So if you look at the picture here we have neuron number 1, and it wants to communicate over here with neuron number 2. And looking closer at the connection piece between the two, so this is going to be our axon terminal of one neuron, and this will be the dendrite, which is the receiving end of another neuron. So looking at it more closely, this would be the axon terminal here of our presynaptic cell. Presynaptic means the before the synapse cell. Over here is the dendrite of our postsynaptic cell. The postsynaptic cell is on the receiving end of the synapse. So the job of the presynaptic cell is to release neurotransmitter via these synaptic vesicles. And the job of the postsynaptic cell is to receive the neurotransmitters. And don't forget there's a space between the two cells called a synaptic cleft. Let's take a closer look at neurotransmitters. So neurotransmitters are the chemical messengers used by the nervous system. So these are going to be the chemicals that are released by that presynaptic cell. The job of a neurotransmitter is to bind to a receptor on the postsynaptic cell. Now typically this receptor is a chemically gated ion channel. So we've studied ion channels in this class before. Chemically gated ion channels are opened via a chemical. So the word before gate tells you what opens the gate, and the word before channel tells you what goes through the channel. So in this picture here, this is our chemically gated ion channel, also known as a receptor in this case. So this chemically gated ion channel, lost my cursor, there it is. When it binds to a chemical, in this case the chemical is the neurotransmitter, when it binds to the chemical, that causes the gate to open, and then ions can flow through the channel according to diffusion. So they just follow their concentration gradient. So these neurotransmitter receptors are embedded in the cell membrane of that postsynaptic cell. So the neurotransmitter binds to the receptor and opens the channel on the postsynaptic cell, allowing ions to enter or exit that postsynaptic cell. That's going to change the voltage in that postsynaptic cell, and we'll talk more about this as we get further into the neurophysiology. The neurotransmitters don't stay bound to the receptor forever, right? They are eventually released, and then sometimes they're actually taken back into the presynaptic cell. We call that reuptake. It's a nice way to kind of reuse the neurotransmitters. And sometimes they are degraded by enzymes. So sometimes an enzyme will tear apart the neurotransmitter. And sometimes the neurotransmitter will just kind of float away. Now let's take a look at neuroglia in a little bit more detail. So neuroglia are also known as glial cells, and these are the helper cells to the neurons in the nervous tissue. They comprise about half of the volume of the nervous system, so there's a lot of neuroglia in your nervous system. Technically, what the neuroglia are doing is they are preserving the physical and biochemical structure of the nervous tissue. Basically, they're just making sure that the conditions, whether they be physical conditions or chemical conditions, they're making sure that the conditions around the neurons are perfect. Right? I sometimes think of them as little slaves to the neurons. They have to make sure everything is perfect for the neurons. And they're essential for the function and survival of neurons. There are 5 different types of neuroglia that we're going to take a look at: ependymal cells, astrocytes, microglia, oligodendrocytes, and Schwann cells. So let's take a look at a picture of some of these things. So this is a microglial cell here, and the microglial cells are sort of like the maintenance personnel of the nervous tissue. They're running around the nervous tissue looking for garbage to gobble up. So they'll gobble up dead and dying cells and any other debris of the nervous tissue. The astrocytes are really important for forming a structure called the blood-brain barrier, right? The BBB. So all of the blood vessels that are feeding blood to your brain and spinal cord, they have a special coating on the outside of them called the blood-brain barrier, and that's made of astrocytes. So the astrocytes get to somewhat choose which things leave the bloodstream and enter into your nervous tissue. Ependymal cells, which we see down here, are important for helping to form a fluid found inside your brain and spinal cord called the cerebral spinal fluid, or CSF. So ependymal cells help to produce this cerebral spinal fluid. The last two microglial cells, oligodendrocytes and Schwann cells, are somewhat similar because they both do the same job. They both form myelin sheaths. So we can see that in this picture here, these blue tube-like coatings that are found on the axons of neurons. These are called myelin sheaths. And basically they're layers of fat around the outside of the axon, insulating the axon. And the myelin sheath insulation is really important for neural activity. It helps the nerve impulse or action potential to travel very quickly along an axon. And it also saves a lot of energy for the nervous system too. We talk a lot more about this in a different video. Let's just compare these oligodendrocytes and Schwann cells. The main difference between the two is location, location, location. Oligodendrocytes form myelin sheaths around axons in the central nervous system, while Schwann cells form myelin sheaths around axons in the peripheral nervous system. But they're both doing the same thing. They're both wrapping themselves around the axon, right? So in the picture down here, we see a Schwann cell forming a myelin sheath. And you can see that the Schwann cell is just wrapping its whole entire self around the axon. So these Schwann cells are pretty much just made of cell membrane. They don't have a lot of cytoplasm, right? So if cell membranes are made of lipids, then myelin sheaths must be made of, if you said lipids, you're correct. So basically, it's a layer of fat. And we know that fat makes a good insulator, right? So these myelin sheaths are excellent insulators. So the oligodendrocyte forms a myelin sheath in just a slightly different way. Rather than wrapping the whole entire cell around an axon, just parts of the oligodendrocyte get wrapped around the axon. That's what we see in the picture here. But other than that, it's ultimately the same. We have a bunch of cell membrane wrapping around the axon, forming this fatty insulating layer. Now, if you look closely at this picture, notice that the whole entire axon is not covered with myelin. So the myelin sheath is choppy, and that is really important. If it was a continuous layer, it wouldn't work right. So there are spaces between myelinated segments. These spaces are called nodes. So the nodes are the areas of the axon with no myelin along them. The areas of the axon that are myelinated are called internodes. The nodes without the myelin are also known as nodes of Ranvier. And again, we talk a lot more about these when we talk more about conduction of action potentials along the axon. But basically what happens is an action potential or a nerve impulse appears to jump from node to node traveling down the axon.